مرحبا بالجميع. Hello everyone. I would like to welcome you to this webinar organized by the Tunisian Académie Beit al-Hikma in partnership with the Palestinian Academy of Science and Technology. My name is Ahmed Abbas. I am a mathematician in Paris and a member of Beit al-Hikma that you can see behind me. Today we have the pleasure and the great honor to welcome Professor George Smith. My colleague, Professor Habiba Mohamed Shabouni, the chair of the science department at Beit al-Hikma, will present in a few minutes. He will then speak for 1 hour and we will take a few questions afterwards. You can either put your question orally in English by raising your hand at the end of the lecture, or you can write your question all along the talk in the chat section, either in English or French or Arabic, and my colleague Amel bin Ammar Al-Gaid will summarize the questions at the end. One last practical comment. So we did a test and just before, and I recommend that you choose the side-by-side mode to see the full slides that George will share. So in French, it's affichage option côte à côte. Now, Habiba, you have the microphone.
Okay, thank you, Ahmed. Good evening, everybody. George Smith is Emeritus Professor at University of Missouri and 2018 Nobel laureate chemistry. And it's my great pleasure to make a brief introduction of our speaker in this very interesting webinar. In 1970, George Smith obtained a doctorate in bacteriology and immunology at Harvard University. After a stay at the University of Wisconsin in Madison, he moved to the University of Missouri in Columbia and remained there for the rest of his career. As he introduced himself at the Nobel Prize Chemistry ceremony in 2018, George Smith belongs to a large scientific community, which includes molecular biologists, protein chemists, evolutionary biologists, immunologists, phage biologists, mathematicians, philosophers of science, etc. Evolution, characterized by diversification, natural selection, and adaptation of species to different environments, has created an enormous diversity of life. George Smith has used the same principles, I mean genetic change and selection, to develop proteins that solve humankind's chemical problems. A common thread of his research has been the study of genetic diversity in antibodies, repetitive DNA sequences, and finally, in combinatorial chemistry. He began to study the assembly of filamentous phages with a specific interest in designing DNA cloning vectors to evolve new proteins. That work led to stunning advances built around his new invention. An elegant method he developed in 1985, the phage display, whereby enormous peptide libraries are generated on the surface of a bacteriophage, allowing extremely rare peptides that bind a target biomolecule, such as a receptor, to be selected using simple microbiological techniques. This method has led to new pharmaceuticals and to development of a range of antibody drugs used as anti-cancer or anti-inflammatory therapeutics. But Smith never patented his discovery. Moreover, he and his colleagues were enthusiastic disseminators of the technology to labs throughout the world, freely providing seed stocks, detailed protocols, and advice. That life philosophy lets me talk about George Smith, the person, as an advocate of human rights and a strong defender of Palestinian rights. Last week, for example, George Smith was one of the speakers of the 3A or AAAS meeting on science, technology, and human rights conference 2020 in a plenary panel on challenges and opportunities for science in the West Bank and Gaza. That aim was to share first-hand perspectives on the situation of scientists in the region and to let participants learn about opportunities for scientists around the world to collaborate and take action. So, behind your screens, please give a warm applause to Professor George Smith for accepting to let us learn more on RNA vaccine for COVID-19. Thank you. And you can start your lecture. George, please.
Well, hello, everyone. I should explain that I missed my plane for Tunis, so I am talking to you from my home on land in Missouri that not too long ago, barely over 200 years, was home to the Osage people. It's a great privilege to be able to talk with you today. And I hope to share with you, let's see, the ideas about RNA vaccines for COVID-19, not only because of their relevance to COVID-19, and this is not about vaccines in general, COVID-19 vaccines in general, but about a particular kind, RNA vaccines, because they illustrate a lot about vaccine development in general. And they're also a very fitting context to talk about development of vaccines and other kinds of drugs in general. So, sorry. Okay, so let's begin by talking about some pandemics that have been brought to us to an end by vaccines. There's the smallpox, the worldwide pandemic of smallpox, which was brought to an end by the first modern vaccine. And that's a process that took about 200 years. The polio pandemic was brought to an end in about 50 years by polio vaccines. But now we're talking about SARS-CoV-2 vaccines that might bring this pandemic to an end in 2 years. This is really warp speed end to a pandemic. I'm going to say first that we don't know that a vaccine will work. We don't know that a vaccine will be important in bringing the pandemic to an end. But I think it's nevertheless really worthwhile talking about the vaccine technology because of general principles that we could learn about fighting pandemics from them. So here's our culprit, what's responsible for this pandemic. And this is the SARS-CoV-2 virion. Virion is the name we give to the actual virus particle that infects cells. So this virus particle, it's about 86 nanometers on average in diameter. And it's like many other viruses, human viruses, it's surrounded by a membrane, a lipid bilayer membrane that's just like and derived from the membranes that surround the cells in our body. In the virus, that membrane is called the envelope. So this is an enveloped virus. And sticking out from that membrane are 50 to 100 copies of a very prominent protein, the so-called spike protein. And the spike protein is essential for the virus to attach to and infect cells. Without that protein, the virus would be completely harmless. And that spike protein is the target of almost all coronavirus vaccine development projects, including RNA vaccines. So that spike protein is a foreign protein to us. And when a foreign protein like the spike protein enters our body, it acts as an antigen. That is, it's recognized as a foreign protein by our adaptive immune system. So adaptive immunity is immunity that is absent in the absence of its target, in this case, the spike antigen, but is induced when that spike antigen enters the body. And adaptive immunity has two major arms. There's so-called humoral immunity that's mediated by antibody proteins that are in the blood and other body fluids. And among those proteins against this spike antigen, I should say that the adaptive immune response is also extremely specific for the antigen that induces it. So for example, the adaptive immune response against the spike antigen doesn't react with any of your other body proteins. It doesn't even cross-react with the spike protein on most other coronaviruses. Anyway, it has two arms. The humoral arm is mediated by antibody proteins circulating in the blood and body fluids. And among them, antibodies against the spike protein can bind to the spike protein on the surface of the virus and block its ability to infect cells. So those are called neutralizing antibodies. So the other arm of the adaptive immune response is cellular immunity. And this is immunity that's mediated by whole cells. And those cells can recognize other cells in your body that have a foreign protein, for example, the spike antigen in them. And it has the cellular immunity has a number of functions, but one of them is to recognize cells that have a foreign protein and kill those cells. And that's another major way that the adaptive immune response protects us against viral diseases. So most conventional vaccines consist of administering the foreign antigen, the spike antigen in this protein, in this case, into the body, where it provokes this adaptive immune response. But I'm going to be talking about another vaccine designs in which it is not the spike antigen itself which is administered, but rather a messenger RNA that encodes that spike antigen. So messenger RNAs, every one of our cells makes tens of thousands of different messenger RNAs. They're made in the nucleus. They're transcribed from DNA in the DNA genes in our chromosomes, and they consist of a sequence of RNA nucleotides that are complementary to the DNA nucleotides that they're so-called transcribed from. And our normal messenger RNAs are equipped at one end with a special structure called the cap, and equipped at another end with a string of about 100 or so adenosines. So in these vaccines, an artificially synthesized messenger RNA is administered. It looks just like our regular RNAs, but the coding sequence is not one of our natural coding sequences, but the coding sequence that specifies the sequence of amino acids in the spike antigen. So that translation of the sequence of nucleotides to the sequence of amino acids in the antigen is carried out by an organelle called ribosomes in our cells, and that process is called translation. So there's a whole apparatus for carrying out the synthesis of proteins, and that's the process of translation. So in this vaccine, when this RNA gets into some of your cells, those cells treat this, this artificial RNA just like it would treat the normal RNAs, and it makes lots of spike antigen protein. So a few of your cells that acquire this RNA become factories for actually producing the actual antigen that induces the adaptive immune response. Now, you can't just take an RNA and just introduce it into the body and hope to get an immune response. And that's because the RNA would be immediately degraded. You have to formulate the RNA in a way that protects it from degradation and mediates its entry into cells. And here is the formulation. These are lipid nanoparticles. They're about the same diameter as the virus particle itself, and they're surrounded by a membrane that's very similar to the membrane that surrounds the virus particle, but it's an artificially produced membrane made from chemically purified lipids. And inside that membrane is one or a few copies of the RNA. That's the lipid nanoparticle. And lipid nanoparticles can be manufactured by a very simple process from 5 highly purified components. There are 4 lipids that are dissolved in ethanol, and then very highly purified RNA made artificially in the test tube, not acquired from cells, and that's dissolved in a watery buffer. And when you mix those 2 liquids in a particular way, the lipid nanoparticles self-assemble in great numbers. This is a very simple manufacturing process. And an important thing about this process is that it's completely generic. It doesn't depend on the sequence of nucleotides in the RNA. Whatever nucleotide might be desired for an RNA vaccine, you manufacture it by the very same process. So what happens when these lipid nanoparticles are administered into the body? Well, they enter into cells, and by a process not shown here, this is item 1 on this diagram, by a process that's similar to the way the viruses enter cells. And lipid nanoparticles end up inside membrane-bound vesicles inside the cell. And inside those vesicles, the RNA is liberated from the lipid nanoparticle and exits from the vesicle. That's step 2 in this diagram. And the RNA is symbolized by these red squiggly figures. And inside the cytoplasm of the cell, they're recognized by ribosomes, and that starts the process of translation. And that makes from every RNA many hundreds of copies or thousands of copies of the antigen. And that's how the antigen arises. Now, I'm not talking with a lot of immunologists here, so I'm going to not talk about the details, the rest of the details in this slide, but I want to summarize them. And that is that another thing that happens when this RNA enters into the cell is that it activates the so-called innate immune response. So the innate immune response is unlike the adaptive immune response in that it is constitutive. It's always present. It's not induced by a foreign antigen. It's also very non-specific. It's not specific for this particular virus at all. Instead, it recognizes generic molecular patterns that are characteristic of pathogens. And one such pattern is an RNA inside a vesicle. In a healthy cell, there shouldn't be any RNA inside vesicles. But if a cell is infected by an RNA virus, like a coronavirus, well, there will be RNAs inside vesicles, and the innate immune system has receptors that recognize that fact and trigger this innate immune system. The innate immune system has a number of functions, but one of the most important is that it's responsible for mobilizing the adaptive immune response. You get a very weak adaptive immune response if the innate immune response isn't triggered. And these RNA vaccines themselves trigger that immune response, that innate immune response. Now, conventional vaccines don't do that. If a foreign protein entering the cell doesn't trigger the innate immune system. So conventional vaccines have to be supplemented with so-called adjuvants. Adjuvants that are added, and those adjuvants trigger the innate immune system. But adjuvants can be toxic. So it's an advantage of RNA vaccines that they themselves trigger the innate immune system. They're self-adjuvanting. So I want to summarize the theoretical advantages of these RNA vaccine design. There's a simple, generic, good manufacturing process from 5 highly purified components, 4 lipids plus the artificially synthesized RNA. There are no potentially toxic components or contaminants that go into this vaccine. A very short manufacturing time scale, easy scale up because it's a simple manufacturing process. And although we may not have thought of this, but the manufacturing infrastructure put in place for a particular pandemic, for example, is immediately usable for future pandemics because of the manufacturing process is completely generic. There's no adaptive immune response to the vaccine itself. And that means it's reusable for other target antigens, a new pandemic. If there were an immune response against the vaccine itself, if you use the vaccine a second time, say for another pandemic, the adaptive immune response against that vaccine might kill the vaccine before it has a chance to induce an immune response. So the absence of an adaptive immune response against the vaccine itself means it's indefinitely reusable for other target antigens. I've already mentioned that it's self-adjuvanting. Another advantage is that the RNA never enters the nucleus and is never reverse transcribed into DNA. And that means there's no possibility of it generating heritable alterations to our chromosomes that may be oncogenic and lead to cancer. A possible problem with vaccines that consist of DNA. So in short, it's a generic, reusable platform that's immediately adaptable to new vaccine targets. And that's a major reason for being very interested in this technology. There are problems with the RNA vaccines. So far, the vaccines need to be kept cold, and that's a really severe problem for vaccination in under-resourced countries, as will be necessary probably if this vaccine is to be used to bring this pandemic to an end. But that's almost certainly a solvable problem. It just hasn't yet been solved. It's very likely that we'll be able to freeze dry RNA vaccines and distribute them at room temperature, and then dissolve them on the site and use them right away. So that, I think that's a solvable problem, but it's not a problem that has yet been solved. So before I leave the subject of RNA vaccines, I would like to discuss a variation on that theme, and that is self-amplifying RNA vaccines. So these are vaccines that are built on the chromosome of alpha virus. That's an RNA virus, like the coronavirus. So it has 2 coding sequences in. The first coding sequence is a long coding sequence that makes a long protein that's subsequently cleaved into 4 smaller proteins that assemble into the replication machinery, the replication complex for this virus. And downstream of that, in the alpha virus itself, is another long coding sequence that is broken up into 4 proteins that are the so-called structural proteins of this virus. These are the proteins that are necessary for the virus particle to be assembled and released from the cell, and the proteins that become part of the virus particle itself, the virion itself. In the vaccine, however, that second part is replaced with coding sequence for the spike protein. And I've put that not in bright red, but in a very faded red, because the second coding sequence can't be translated from this RNA. Our ribosomes only translate the first coding sequence in line, but that's not a problem, as we'll see in a minute. So what happens when one of these self-amplifying RNA enters the cell in the form of a lipid nanoparticle? Well, the first thing is that first coding sequence is made into the protein, which is processed to make the replication complex, which I'm symbolizing in this way. So what the replication complex does, it's responsible for replicating the RNA. So it uses that incoming RNA as a template to make a complementary RNA that goes in the opposite direction and that has complementary bases. So where there's a U in the vaccine RNA, there's an A in the minus strand, the so-called the complementary minus strand, etc. That's the complementary RNA. It has the same information as the vaccine RNA, but it's in the reverse order and using the complementary bases to each base in the vaccine RNA. And that same replication complex turns around and uses the minus strand RNA to make many more copies of the vaccine RNA. So you started with 1 vaccine RNA and soon you have thousands. And the replication apparatus also uses the last third of the minus strand RNA as a template for making a shorter RNA, a so-called subgenomic RNA, where that second coding sequence is the first one in line and can be translated. And in the case of the vaccine, that second coding sequence is for the spike protein, the desired protein. And I should mention that no virus can result from this, because the necessary proteins, the structural proteins that are necessary for making an actual virus and infecting another cell are absent. So this can't be transmitted to other cells by infection, which would be a very dangerous vaccine indeed. So there's a key advantage of this kind of design, and that's because a single particle delivering a single RNA into a cell results in many thousands of spike messenger RNA molecules. That synthesis of RNA molecules is sustained because even though RNA molecules are eventually break down in the cell, they're constantly being renewed. So there's a sustained but not permanent expression of the spike protein over a period of several weeks. And that holds out the possibility of a single dose vaccine, just a single dose being required instead of 2 doses, as is probably going to be required for non-self-amplifying RNA vaccine design. There are potential problems with the self-amplifying RNA vaccines. We've already talked about the problem that they have to be kept in the cold, but as I've already described, that's probably a soluble problem. But there is a special problem with self-amplifying RNA vaccines that's not present in the non-self-amplifying RNA vaccines, and that is that in this case, there is a potential for an adaptive immune response against the vaccine itself. So it's possible that this design will not be indefinitely reusable for new vaccines against new targets. We don't know about that yet. So before I move on, I would like to define roughly 2 phases in the creation of a new vaccine, and indeed any new pharmaceutical. The first phase is discovery and technological innovation. This is exploratory science driven by curiosity and professional ambition. It's overwhelmingly carried out in academic labs with public funding. The innovations such as RNA vaccines that emerge unpredictably. The innovations emerge, such as the RNA vaccines, emerge unpredictably from global scientific communities, not individual researchers or research groups. So the other phase is, I'm calling development. I should say these phases overlap somewhat, but it's a pretty good overall way of describing the creation of a new pharmaceutical, including a vaccine. So development is carried out by corporations driven by profit. This is mostly responsible for the final optimization steps of of an innovation, and for the human trials, not for discovery and innovation. It's privately financed with the promise of government-granted patent monopolies if a successful drug, for example, the vaccine, results. And this means that the corporations that carry out this development must have key patent rights. And those patent rights are mostly purchased from public institutions or from startup companies that are associated with those public institutions. And in order to facilitate the systems, governments now permit researchers and their institutions to pursue patent rights to discoveries and innovations that arise from publicly funded research. This hasn't always been the case, but over the last 40 years, pioneered by the United States, that has been the case. So let's talk about the progress in the development of an RNA vaccine for SARS-CoV-2. The discovery and technological innovation phase for RNA vaccines is pretty much finished by the year 2000. It's been around for almost 2 decades. But development, in contrast, is barely started when this pandemic hit. So there were generic issues yet to be resolved. Can the vaccines be freeze-dried and distributed at room temperature? Almost certainly they can, but the development for that never took place. Are there generic safety issues for this particular vaccine design that we didn't suspect? We don't know that yet. Do these vaccines induce a vigorous adaptive immunity to the target antigen, both antibodies and immune cells? We don't really know that yet. And in particular, we don't know how long that adaptive immunity lasts. We just don't know that. And that's a generic question about this vaccine design in general. And in the case of self-amplifying RNA vaccine, as I've already mentioned, we don't know whether adaptive immunity to the vaccine itself limits its reusability. These are all generic questions that were not answered before this pandemic hit. Of course, there are also SARS-CoV-2 specific issues that couldn't be answered until we knew what we were up against. And those are issues to be resolved in human trials. Are there safety issues that are specific for this particular antigen? We don't know that yet. And that's what's currently being investigated in the current human trials. And is vaccine-induced adaptive immunity to this particular target actually protective against disease. We didn't know that, and we couldn't know that before we knew what particular pathogen we were up against. But we did know a lot about the generic issues, and they weren't answered before this pandemic hit. And that means that we were faced with an emergency development problem. And that development has been a fauda. For my Palestinian colleagues, I don't mean this kind of fauda. So let's just talk about what a mess, what a fauda this emergency vaccine development has been. So as I've already mentioned, the RNA vaccine platforms were ready for development 20 years ago, but there's been no market incentive for corporations to explore generic development of these platforms in preparation for a future pandemic, because no product, only information results from this, no product that can be sold. So this would be an extraordinarily risky investment for a corporation. And that's resulted in very hasty, not well thought out investments on an emergency basis. And I just like to talk about one of them. Our country, the US, has made a very hasty investment in a company called Moderna for its RNA vaccine. We've invested almost $1 billion of public money for the development of the vaccine by Moderna. We've also committed to buy 100 million doses at inflated prices, about $15 per dose versus about $4 per dose production cost. And we have an option for 400 million additional doses. But we've allowed the company to still protect its commercial secrets from other companies, so they still will have patent rights to this. And by the way, this vaccine is probably only going to be available for US citizens. And I should just say that the emergency development in other capitalist countries has been pretty much equally chaotic. Could we do better than this? There are a number of economists who argue that we can do much better for developing vaccines specifically and for developing drugs in general. And that's what I'd like to spend my remaining minutes explaining. So I'm start by saying that governments today are very averse to collecting taxes. And that means that governments that in countries that develop new pharmaceuticals prefer to stimulate the development of those pharmaceuticals by the promise of government-granted patent monopolies rather than by direct government spending. And as a sideline, I should say that there are many countries who are whose governments are tax averse, not because they want to be, but because they're forced to be. So for example, governments that receive loans from the International Monetary Fund are often required to adopt very severe austerity measures that they don't willingly do and that are run against the will of the people. Well, the result of this kind of funding of drug development is that we pay monopoly prices for our drugs. So in the United States in 2016, we paid about $450 billion for pharmaceuticals. That's well over $1,000 per person in the United States. And other countries also pay monopoly prices, and that's because the World Trade Organization, the World Trade Organization forces countries all over the world to recognize patent rights from companies in the United States and elsewhere. So other countries are also forced to pay monopoly prices for drugs. maybe not quite as much as we do in the United States. I would like to also highlight another cost, and this is the cost that I'm calling misery from monopoly prices. And this is an attempt to monetize the fact that some people can't afford to pay those monopoly prices, and society isn't willing to pay the monopoly prices for them. They have to go without those drugs. And this is the economists' attempt to monetize that suffering. I don't think we should take that value, that monetary value, very seriously, but this is certainly a real cost to society. And it reminds us that drugs aren't just a commodity, they're also a public good. And the development of drugs is not merely a matter of commercial prosperity, but also of basic social justice. Anyway, an alternative to this that would avoid these costs would be to publicly fund the development of drugs instead of funding them by patent monopolies. And it might surprise you to learn that we wouldn't have to pay that much in the United States, for example, for publicly funded drug development. We wouldn't have to pay that much for drug development. Only about, it's estimated about $75 billion in 2016. That was the amount of money that pharmaceutical industries spent actually on drug development, all the human trials, et cetera. The rest of their money is spent in other ways. Of course, so let's ask how this public funding would work. Well, presumably it would be in the form of contracts, maybe long-term contracts to companies in broad areas. These contracts would ask companies to do drug development in broad areas of concern. For example, for vaccines for future pandemics. All the results of their development would be immediately made public, both positive and negative results, so other people can use them. No patents would be allowed on the resulting drugs. They become generic. Any company would be free to manufacture and sell the drug at a market price. And as a corollary of that, patents would no longer be allowed on the results of publicly funded discoveries and innovations either. And the performance of companies holding these contracts and their chance of getting their contracts renewed or continued would be assessed on the usefulness of their information, of their results to society. And that way, many perverse incentives would be avoided. And I'd like to cover a few of those perverse incentives. If you're a corporation driven by profit, don't develop cures or vaccines. Concentrate on lifetime treatments, guaranteeing a steady revenue stream. If you're a corporation driven by profit, it's a good idea to try to capture market share with Me Too drugs with no benefit to society. Again, guaranteeing some extra money. If you're a corporation driven by profit, don't prepare for future challenges like a pandemic. That's a way too risky investment. If you're a corporation driven by profit, hide your results, especially your negative results from regulators, and your positive results and your negative results from your competitors. These are all incentives that are avoided if we publicly fund drug development instead. Of course, we'll still all have to pay for the drugs, but we'll have to pay for them at a free market price, not at a monopoly price, because companies are free to get into the market for manufacturing and selling these drugs using the information that arises from publicly funded drug development. So we save some money that way. And this is a graph of the money that we would save, subtracting the red expenses from the blue expenses. That's the money we save. So the bottom part is kind of like virtual dollars we save. This is the dollar value placed on the misery we avoid to society from monopoly prices. But as I've emphasized before, this misery, maybe we shouldn't take the dollars seriously, but the misery we should take seriously. That's a genuine social cost. So the upper part are real dollars that don't have to be spent on drugs, dollars that would have been used for patent lawyers, for advertisers, for lobbyists, and especially extra earnings for wealthy investors. Now, the loss of jobs for patent lawyers, for advertisers and lobbyists, those are real economic losses. They'll have to get other jobs. But I think that many would agree that the loss of those extra earnings for wealthy investors might be an advantage rather than an economic loss. Because after all, patent monopolies are an important factor in the upward mobility of wealth in our societies, an important contribution to the obscene wealth inequality that characterizes capitalist countries today. And in that sense, avoiding them would be a great advantage. So that is my vision for a new way of developing drugs. I don't think it's very visionary because it really builds pretty straightforwardly on the way we fund discovery and technological innovation already. And I think it aligns better with our widely shared ideas about social justice. Thank you. I'm going to stop screen sharing.
Thank you. Okay.
Can you, I'm not able to stop share. Oh, Sarah. Yeah.
Oh, okay. Is it okay? Thanks a lot. Thanks a lot, George, for this wonderful lecture. So we will take now a few questions. So there are a lot of ideas and on at least two aspects of the problem. We will start by taking oral questions. So please raise your hand if you'd like to ask your question. And I would like to kindly ask you to be extremely brief to allow as many questions as possible. So you have 1 minute to ask your question. So I see a first hand raised by Leila Ammar. Okay, so you can ask your question. Does it work? Yes, you can ask your question. So please activate your microphone.
She's still muted.
Yes, you have to activate your microphone. We don't hear you.
Leila, you have to unmute your microphone.
Okay, so we will come back to Leila later. Let's go to the next question. So the next question from Laura. So Laura, you can ask your question. Please unmute your microphone.
Hello, can you hear me?
Yes.
Yes.
Thank you for inviting me to this very interesting event. I am a director of science with a basis in molecular biology, so I would not ask questions on the economic part, which was indeed very useful and interesting, but on some details of the reverse transcription, because it was very interesting to me, and you didn't have time enough to develop why we can be sure that this will never happen. In the self-amplifying, I heard that there is a part where there is a codon-anticodon step. So it is kind of reverse transcription. So the double-stranded RNA somehow exists in a moment. How you can assure that this will not go up to the DNA? So this is the first question. Then I have another, and that's finished. The second is that in the introduction, you said that no potentially toxic components exist in this new form. But then we were questioning at the end whether antigens, so the spike, is it toxic itself? So it doesn't exist at the beginning, but it might be the result. So those are the two open questions from my side. Thank you.
Okay, so it's true, Laura, that that the replication for self-amplifying RNAs, but not for the non-self-amplifying RNA vaccines, that the RNA during replication becomes double-stranded, at least in short segments of the two molecules, of the parent molecule and the molecule that's being made from it. But this is not reverse transcribed into DNA, and also there is no process that gets these RNAs inside the nucleus. So the possibility of changing chromosomes doesn't seem to be there. Maybe you're thinking of something that I'm not. And I will add. Oh, sorry. What was the second one? Laura, are you back? Are you there?
Okay, we will come back to her later. So
yeah, I'm sorry.
Laura, raise your hand again. So I will give you the question at the end. So we move to the next question from Professor Ahmed Bouazi. So please activate your microphone.
Professor, thank you very much. I am a physicist. I am not a biologist, but I understand today how things are going to make this new vaccine. And I thank you very much for explanation, and thank you very much also for your mind, your social mind, that we need, all of us, we need to have drugs, to have vaccines, not to pay so much as we are doing now. Even here in Tunisia, we are paying a lot of money for drugs. Thank you very much. I was really happy to attend today your conference.
Well, thank you very much, Ahmed.
So we go back to Laura. So we are back. Could you ask?
I'm sorry, I forgot your second question.
Thank you. I will be fast. I don't want to cover all time. So the second was, there is no toxic components from the beginning, but the result might be toxic since we cannot yet know about the antigen, the spike protein's effects.
Laura, you're absolutely correct. And I tried to say that when I was talking about problems we don't know specifically for this particular target, the spike protein. We do not know whether, for reasons we can't anticipate, having spike protein made in some of your cells might lead to unexpected problems. We don't know that. But we can answer whether generically this happens all the time with the RNA vaccines. And that seems very unlikely because the actual vaccine itself doesn't contain toxic components. It would have to come, presumably, from the protein that's made as a result of the vaccination, not from the vaccine itself. But that's a very good question. And of course, it's one that has to be addressed for all vaccines and all drugs. You have to actually test whether it's safe.
Okay, so we will see if Leila is able to speak. So Leila, could you ask your question? You raised your hand. So could you activate the microphone? Leila Ammar. Okay.
Hello.
Yeah,
hello, Leila.
Excuse me, I didn't follow continuously your conference because I was driving.
Ah,
So
okay.
I haven't any question, but I understand that it was a very strong and helpful, interesting conference. And thank you for this.
Okay.
Well, you're very welcome.
Okay, so before giving the microphone to Amel, Habiba has a question. Go
Yes,
ahead.
because I can't have my hand on the screen. Just a question about, is there any possibility to RNA to be interacted with mitochondrial DNA in the cytoplasm?
With interact with DNA.
With mitochondrial.
Or mitochondrial DNA.
Mitochondrial.
I don't know. I guess it's a possibility it could get into, in the RNA could get into a mitochondrion. I don't know a route for that. And I don't know what kind of interaction would be possible, but I should point out again that we don't know of any route for that RNA to be turned into DNA, and a DNA interacting with the DNA, that does have the possibility of making heritable alterations in the target DNA. So, it's possible, but it doesn't seem very likely.
Okay, thank you.
Okay, so we have a question, I think, from Professor Arbi Bougara. So, Professor Arbi Bougara, you have the microphone. You should activate your microphone. Please activate your microphone. Yeah, could you please activate your microphone? Could you put your microphone? I don't think he's able to activate his microphone, so we will see. We will come back to him later. So now we go to Amel Ben Ammar Al-Gaid, who will summarize the questions on the chat section. So Amel, please activate your microphone. Okay.
Yes. Do you hear me?
Yes, we hear you.
Yes. Thank you very much for this conference. It was very, very interesting. And we will have, I think, 4 kind of questions. The first is around the choice of the antigen, spike protein. Do you think it is the better antigen to choose to generate the best T-cell response against the virus? Because, as you know, some studies have showed that T cells and TCD8 cytotoxic cells may be generated, I think, against N antigen, M antigen, and more antibodies are generated against spike. So, as you know, viruses generate T and cellular immune response, and that are very, very efficient against virus than antibodies. This is the first question.
Okay, let me answer that question.
Mm-hmm.
We have yet to know really whether in the vaccine design, whether we get a cellular immune response against the spike protein. And you've mentioned you've called these T cells, and I should explain to people that T cells are the name given to the cells of the cellular immune response. And she mentioned one particular kind of T cells, CD8 plus T cells, and those are the cells that are able to recognize a cell having a foreign protein and killing that cell. So, and she also mentioned another antigen from the coronavirus, the N protein, the nuclear protein that's inside the virus. It's not exposed on the outside. And indeed, you can generate an immune response against that N protein. And there are other proteins as well, but that's the most abundant protein. And so it is in some ways an attractive target. And some vaccine designs, not as far as I know any of the RNA vaccines, but some vaccine designs either consists of the N protein or combines the N protein with the spike protein. But I don't know if it's true, if we really know that we don't get a cellular immune response against the spike protein when it enters in this manner as a result of an RNA vaccine.
Mm-hmm. Okay, so thank you. The second question is around safety of RNA vaccine comparatively to the other type of vaccine that are developed now. And you have 10 kind of vaccine that are now being developed. Why do you think that this is the safer vaccine that we could use it?
Well, that's a very good question too. And I don't think I want to make the claim that this vaccine is inherently safer, so much as I'd like to make the claim, as the proponents of this do, that it's much easier to make it safe because the vaccine components themselves are highly purified artificial components that have very little prospect of being toxic in themselves. But that doesn't mean that you can't make a safe vaccine with other designs. So for example, the Chinese are developing and are pretty close to, looks like they're pretty close to actually deploying a vaccine that is a very old-fashioned design. You take the virus and you treat it so it's inactivated, it can't infect, but it still provokes an immune response, and that's what the vaccine is. But to produce a virus, that has to be done in cells. Anytime you're using a living system to produce the vaccine, you have to make sure that you're getting rid of the components in the living system that could be harmful. So it's hard. It's a hard thing to do. So investing in RNA vaccines, if they work, safety would be a generic property of them, not something that has to be worked out with each new vaccine target. So I think that's the claim that I would make in terms of safety for these vaccines.
Thank you. For self-amplified RNA vaccine, you think that it should be less expensive?
Yes, I think it will be less expensive. And of course, it will be much less expensive if we don't pay monopoly prices for it. And the major developer of the self-amplifying RNA vaccine at Imperial College London, his name is Robin Shattuck and his group, have made provisions for manufacturing their vaccine. They haven't yet embarked on large-scale human trials, but they've made arrangements for this vaccine to be produced at cost and sold at cost in under-resourced countries and mid-income countries, though probably at a higher price for rich countries. So yes, it would be cheaper to manufacture, and it might be much cheaper if the whole problem of the cold chain, you know, having to keep the vaccine cold, if that's solved, then it could be much cheaper, because there keeping the vaccine cold is not a major, would not be a major expense.
Mm-hmm. The problem is for temperature. If you have proteins or RNA, you have problem because with temperature, you know, to have stabilization of the product. If you had DNA as vaccine, the problem will be, you know, solved because DNA is more stable and you don't need special temperature to have it okay.
Okay, so that's a question
but
about
the problem for.
Oh, I'm sorry. Go ahead. Continue.
Is it the first question, or it's
the problem for the problem? Do you think that if we can solve some problem of safety for DNA vaccine and using DNA vaccine and solve the problem of temperature?
Well, that's a good question, and I should say that I am not an expert in vaccines in general, although have been very interested in them for many years. So I'm not entirely qualified to answer that question. But about the stability of RNA, RNA is the most conspicuous way that RNA is unstable is that it's degraded by enzymes. But the manner of manufacture of these RNA vaccines, there aren't any enzymes around. I mean, the RNA that's made in the test tube is made in a way that there are no enzymes around except the enzymes that are necessary for making the RNA, the one enzyme that's necessary for making the RNA. And the manufacturing process that uses that RNA also doesn't have any enzymes around. RNA is also unstable in other ways. It's certain, like DNA, it's unstable to light. It's unstable to acid and base, but these are avoided in the manufacturing process. And I think that in freeze drying an RNA vaccine, this would really pretty much bring to an end any of the, as long as you keep that dose out of the light, this would pretty much put an end to the major destabilization processes. And it doesn't have to be rock solid stable. I mean, if you can distribute it within a few weeks, it's very likely that the RNA will last that long, because regularly in the laboratory we keep RNAs around for that long, even at room temperature, as long as you get rid of enzymes.
Mm-hmm. And finally, it's about patents. As you know, countries like Tunisia and other poor countries cannot pay even for some drug that have been developed these last years because of the cost. And the regulation of the price of new drugs and vaccine should be discussed really at a global level, because we in such a way we are going to exclude a lot of people from some new drugs, and we'll have a medicine at 2 speeds. And I am very happy that you are very sensitized and aware from the problem, because in our country, we know that this is a problem. We cannot say to people, you cannot use this drug because your country cannot pay for it, or because it is too expensive, such as, for example, antibodies against checkpoint for cancer, for example. And until now in Tunisia, we cannot use such antibodies. So the general, vision that you gave, could you think that we would have a large discussion and decision at global level about this, and involving not only companies but government and also the civil societies?
Well, you practically gave my lecture for me. So, yes, I mean, I'm fully for that. And also, Tunisia, scientists in Tunisia, maybe Tunisia is poor in money, but I don't think poor in human ingenuity. So, Tunisian companies would be free to compete in the market for manufacturing these drugs. Maybe they could become a supplier of some really important drugs. And we in the United States would benefit too, because we would be getting drugs cheaper, maybe cheaper than any country in the United States could manufacture the drugs for. And that would be a way of spreading the economic, the raw economic benefit of drug development more equally around the world. So it's not just in having cheaper drugs, but also in having the ability to compete in a fairer marketplace that doesn't give all the benefits to rich countries.
Okay.
Okay, so is it over, Amel?
The last question is about
Okay, so brief, because we still have some questions.
The memory against vaccine and the time that we will need to decide if the vaccine is really, useful. And do you think that the trial that are being developing and now will give their answer when? In 1 year, 6 months, 2 years? What do you think? Before having this vaccine on the market, how many time do we need?
Well, the trials that are currently underway are, the results are continually assessed as the trials continue. And the idea is that when the regulatory agencies are satisfied that the vaccine will give at least 50% protection against getting infected, and are also satisfied that there are no immediate side effects, ill effects of the vaccine. Once they're satisfied, then they'll authorize beginning to roll out the vaccine and actually use it. That's the way that these vaccines are being, these vaccine trials are being conducted. Now, of course, those vaccine trials, and really this is true of pretty much all vaccine trials, never are able to assess rare long-term ill effects, side effects that don't appear in the early days. We just don't know that. And truthfully, that's something we don't know in general for vaccines. We also don't know, we will not know from these trials the duration of the protection that the vaccine gives, whether it's 2 months or a year or whatever. We just won't know that from these trials. So it will be potentially, it will be rolled out before we have answers to those questions. Of course, once it is rolled out, then we will, that's a massive, like you could say, a phase 4 trial that could give us those answers. And that, I think, is pretty similar to vaccine development in general.
Okay, so thanks, Amel, for summarizing the questions on the chat section. We still have 2 questions from Silarbi Bougara and Shawqi bin Abdeslam. So Silarbi, you can ask your question. So please, could you unmute your microphone? We should unmute your, yeah, please.
Okay. Thank you very much, Professor, for this beautiful and unforgettable lecture. Thank you a lot. Let me tell you that I often write here in the Tunisian media about your struggle for the justice and for the Palestine. Thank you a lot. Now, I am going to ask you maybe a question which was made by Amel already. I'm sorry. You spoke about the obscene profits of the made by the business and by the patent related to drug and vaccine by the multinationals. This profit, as you know, are killing people in the third world and in our part of the world. But Fleming got no patent for his penicillin discovery. That my question is, what happened since that time? What happened to the values and ethics of the European and American? Thank you very much.
That's such a good question. And I should say that until about 40 years ago, that was pretty much the norm. You know, like vaccines weren't considered a drug that you made profits from. They were a public good. But 40 years ago, and that was also true for many pharmaceuticals. But 40 years ago, the United States led in a campaign to allow publicly funded research to innovations that emerged from publicly funded research to be patented by the researchers themselves or by their institutions. And as I tried to explain, that was an attempt to get companies to put their own money in drug development, including vaccine development. So that is that's a change that's taken place, and it's taken place in the context of a general switch to neoliberal economics that's taken over pretty much the whole developed world. So we have switched to a system that is where we don't rely so almost exclusively on the so-called free market for our technological advances, to a system where we do, and pharmaceuticals fit into that model. So that's I think what's behind the high prices that we're going to pay for vaccines that the first world really wants to develop, and also by the same token, why we don't have vaccines against malaria and other diseases that are characteristic of poor countries. And this would be, you know, from a social justice point of view, of a vaccine against malaria would be just a great advance. And we don't, there's very little money that's put into that. It's not zero money though. People are trying to do it, but it's a really hard project to get companies interested in investing in.
Okay, so we still have 2 questions. So Shawqi bin Abdeslam, please could you activate your microphone? Okay, you go ahead.
Yeah, okay, thank you. Nice and great talk, George. Thank you so very much. And I want to thank the organizers. Just a simple and fundamental question. Is there are any animal models to answer whether adaptive immunity could inhibit reusability of RNA vaccine right now?
Yes, RNA animal models have been used. They're being used for this new SARS-CoV-2, but they were also used for in an attempt to develop vaccines for SARS, which is closely related coronavirus. And those models have been very promising. They've given promising results, but we should just remember that drugs and in general and vaccines in particular that are successful in animal models don't necessarily work when they're applied to humans. But these wouldn't be being pursued so vigorously if there weren't really promising results from animal models. And the SARS, research on development of SARS vaccine really pretty much came to an end when the pandemic came to an end in ways that didn't have to do with the vaccine. And it's, I think if we'd had this publicly funded model of drug development, that research would have gone on in ways that could answer the generic problems, even if it wasn't needed for a pandemic that went away. So that's an example of the kind of incentive that would be present in public funding that is absent in patent monopoly funding.
Thank you. So we will close by a question from Palestine. And now we have Ibrahim Shalail, if I pronounce well, sorry. Could you activate your microphone?
Yes, is it okay?
Okay, yes. Okay, so sorry.
Yeah. So first,
your name, please. Sorry, I mispronounced.
Ibrahim
Okay.
Shalail. So as you mentioned, I am a Palestinian. Let me tell you, Mr. Smith, that the kufiya suits you very, very, very well. We do thank you. My question, during the discussion, you mentioned the RNA and the protein. So I would love to know your idea. As you know, in the community studying the origin of life, we have debates going on whether the RNA came first or the metabolism first. So was it the egg or the chicken that originated the life? So I would love to know if you have an imagination or an idea about whether it was the RNA or peptides and so on.
Well, I wasn't prepared for questions in poultry science, but I will. So this is really way far from my area of expertise. My understanding is that there's a pretty broad agreement, not universal, that that the first sort of replicating type of life, pre-life forms were based on RNA, but so there's an RNA world, and we have remnants of that hypothetical RNA world still with us. So, for example, the enzymatic functions of ribosomes are carried out by their RNAs, not the proteins that are associated with those RNAs. So that's the prime example of an enzymatic activity that resides in an RNA, not in a protein. Of course, nowadays, almost all other enzymatic functions are carried out by protein enzymes, but that certainly is suggestive that there was an earlier time when biological catalysts were RNA. So I think that I'm just not I don't know the objections to this view. So I'm absolutely not an expert, but it seems to me, from my really novice point of view, that there's a pretty good argument for the RNA world preceding our protein-based enzymatic function present living systems. As for other metabolic activities, of course, the RNAs had to be assembled in some way or another. In order for them to replicate, they had to be templated in some way. And all these are questions that I'm pretty sure others have addressed, but I am really not qualified to answer.
Okay, so thanks a lot.
I just ruined my reputation, you know. I'm ignorant in many fields, and that's one of them.
So thanks a lot, George, for this wonderful lecture, which brought a glimmer of hope in this in the heart of darkness. So unfortunately, we have to stop here, but the video and I hope maybe also the slides will be shared on the website of Beit al-Hikma. I will also send them to all participants. So I would like, first of all, to thank our partner, the Palestinian Academy of Science and Technology, and all participants, especially our colleagues from Palestine. And we say thanks again to George for this wonderful lecture. Goodbye and stay safe. Goodbye.
Thanks again.
Goodbye, everyone.
Goodbye.